Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the lecture series on food microbiology. Today we are going to discuss about the various factors which control microbial growth in food. Microbes, as you saw, are everywhere. And when we talk about food, the plant or the animal sources are the major sources of food, and these plants as well as animals can be infected by microbes. So, in nature, the plants and the animals which serve as food source have evolved mechanisms of defense against the invasion and proliferation of microorganisms. Which means that, for example, in the case of plants, we have so many metabolites which are, or otherwise we call them as phytoalexins. The phytoalexins, you have so many lectins, so many secondary metabolites of plants which are secreted by the plant to prevent infection. Like that, animals also have their immune system which will adapt and which will fight against microbial infection. So, whatever these plants and animals in nature have evolved, that quality would also be, that effect would be retained in the fresh food. So, whatever the living animal or the plant already had, remember, all plants food are still living food. Animals get killed, but then the quality, that immune system which had adapted to fight against that microbial, that would be retained in the fresh food. So, if we know the natural phenomena, this can be used to prevent or retard the microbial spoilage of the products derived from them. Meaning, if we know that in this particular plant or in this animal, this is the particular sort of immunity which has been developed to fight against this particular microbe, then we can utilize that as a strategy to preserve the food which has been derived from that particular plant or that particular animal. We have what we call is intrinsic factors and extrinsic factors which control the growth of microbes in any food. Let us see the intrinsic factors first. They are the pH, the moisture content, the oxygen reduction potential, the nutrient content, the antimicrobial constituents, and the biological structures. We will see how each of these factors, the 6 factors, control the microbial growth in a particular food concerned. Let us take pH. The most microbes grow at pH values around 7, which means neutrality. That is, if you consider, we say that a pH of between 6.6 and 7.5 is ideal for microbial growth, and you have very few organisms which grow below 4. But then we also saw the acidurics which can grow at less than 4. Depending upon the type of microorganisms, as we saw, the ideal is 6.6 to 7.5, which means that if you change the pH of the food to less than this or more than this, you can control microbial growth. Most of the bacteria are very fastidious in their relationship to pH, with the pathogens being very, very finicky or very fastidious, meaning beyond, if I say this 6.6 to 7.5, this would be the ideal. So, either less than that or more than that, the bacteria will naturally stop their replication or stop their growth. So, preservation becomes easy, preservation of food becomes easy when you change the pH, because the bacteria being finicky will not grow. And among the bacteria, the pathogens are the ones who are very strict. Even if you slightly alter the pH, they die. So, the contamination of food by foodborne pathogens becomes easy, because if you know the pH which is ideal for their growth, and you alter the pH of the preserved food so as to prevent their growth. Now, In the case of lactobacilli, remember pH minima of certain lactobacilli is dependent on the type of acid which is used and the preservation which is used. For example, citric acid, hydrochloric acid, phosphoric acid, tartaric acid, etc. permit the growth of the lactobacilli at a lower pH value. Whereas if acetic acid and lactic acids are used, the lactobacilli proliferate at a higher pH. So, depending upon the acid which is used as a growth substrate, then the pH minima of the organism can be modified. Let us consider fruits. Remember, when we talk about fruits, fruits are actually formed in the plants, right? And what do you call a fruit? Is it because it is green, or is it becomes ripe? In both the cases, what you call you have what you call, suppose when you are going to do the harvest of a fruit. For example, if I say mango, you have an unripe green colored mango versus an orange colored ripe mango which emits this flavor or the aroma of the fruit. When you take an unripe mango, green colored mango, if you cut it and eat it, it is going to be sour, right, which means acidic. Now, the same green mango, when it starts ripening, okay, the green color changes, it breaks down, you are getting the orange red, which means that the chlorophyll has broken down. Now it is sweet because acidity has reduced, it is now becoming more of glucose, the sugar content. The carbohydrate has now become more of sugar, which means that the pH has changed. So, when the pH changes, along with it, the microbes which can infect also get changed. Okay? So, now acidity has reduced here. So, what happens here? What happens is that the mold and the yeast can cause spoilage here. So, and even in acidic conditions, most of the mold and the yeast can grow at pH values which is less than 3.5. So, in the case of fruits, the spoilage is mostly caused not by bacteria, but mostly by the mold and the yeast, because bacteria cannot grow there, whereas these can grow. Seafood. Seafood, here an ultimate pH of 5.6 and above is what makes it safe. So, at a pH beyond the 5.6, then both the bacteria as well as mold and yeast can happily proliferate in the seafood, destroying them. So, for preservation methods and processing methods, the pH would have to be modified either less than that, because more than that it is going to get spoiled. Then vegetables. Vegetables normally have a higher pH content than fruits. So, vegetables are more prone for bacterial contamination, bacterial spoilage than fungal spoilage. Now, you have some foods which have an inherent pH, acidity. This inherent pH or acidity, now where does this inherent pH come from or inherent acidity come from? This is because of the microbes which are present in them. So, food already has microbes. The presence of this microbe causes a particular acidity. Now, if we change this acidity or the pH, those microbes will die. So, by changing the pH, you can kill the inherent microbial organisms which are present. So, this inherent pH, which is because of the microbes, is called as biological acidity. This is what you see in fermented milk, sauerkraut, pickles, etc. The acidity caused because of the microbes which grow and proliferate. They are not destructive in the sense that the flavor, they are not they spoil, but at the same time they are not pathogens. So, this is the biological acidity. And here the source of acidity does not matter, because of the acidic nature, the keeping quality, shelf life, the preservation methodology, the food is preserved, because even though the biological acidity is there, the other microbes which can spoil the food cannot proliferate because of this inherent acidity. So, this becomes a food preservation technique. When some food are able to resist changes in pH than others. So, when you drastically change the pH of a food, the flavor can become a distaste. The flavor can change, the nutritional composition can change. So, then it can become spoiled. Some food types can resist such changes. So, that type of food we call it to be a buffered food. Okay. Now, for example, meat is more buffered than vegetables. This buffering capacity in the meat is because of the type of proteins which is present in the meat. The animal products have a different type of proteins than what is present in a plant product. So, this type of animal proteins can resist changes in the pH and can have a better keeping quality So we tend to say that it is buffered. Now when microbes grow, as I said, during the growth of microorganisms, the pH, the acidity of the food changes. So the quality of the food will also change. So if the food is buffered, it would be able to resist this change. Whereas if the food is not buffered, it will get spoiled. So if the natural or the inherent acidity of the food, especially fruits, have evolved as a mechanism of protecting the tissues from destruction by the microbes. Now, let's now see how this happens. Fruit, as you know, are reproductive part of the plant. The plant produces fruit so as a way, because the seeds which are inside the fruit are the reproductive structures, it will germinate. Now, to protect what do you require for the seed to germinate? One, the fruit has to be taken away, it has to be distributed into far areas. So for that, you require microbial dissemination, as well as at the same time you don't want it to be destroyed before the seed is ready to germinate. So for protection, you have these acidity, the inherent acidity in the food, so that the spoilage organisms do not destroy it before it is mature enough. And also secondarily, when the spoilage microbes do not destroy the fruit per se, the seed which is inside the fruit is also protected. You are not having disruption. The adverse pH affects, there are two aspects of a microbial cell. How does a change in the pH kill the microbe or how does it affect the microbial metabolism? There are two different aspects here. One, the functioning of the enzyme and the transport of nutrients. The cytoplasmic membrane of the microorganism is normally it is relatively impermeable to hydrogen and OH ions. Their concentration in the cytoplasm therefore remains reasonably constant in spite of the wide variations that occur in the pH of the surrounding medium. So what we are trying to say is, we already know that enzymes have a specific pH in which they work. Like that, on a microbial system, on the bacterial membrane, you have transporters which transport hydrogen ions, hydroxide ions, chlorine ions, sodium ions, potassium ions, etc. Each of this, and you have what you call as the membrane potential, which is maintained because of the concentration of this H plus and the OH ions. When this membrane potential becomes disrupted, the transport of nutrients becomes affected. At extreme stage, the bacterial membrane will become lysed, destroying the bacterial cell. So, when the microorganisms are placed in environment which is either below, above or above neutrality, then their ability to proliferate depends upon their ability to bring the environmental pH to become more optimum value or change. So, when the organism is placed in an adverse pH, for survival, it will try to change its metabolic pathway and secrete substances so as to alter the pH to a more conducive type where it can grow, it can sustain, it can replicate. For example, when you place an organism in acidic environment, the cell will either keep hydrogen ions from entering or it will expel hydrogen ions as soon as it enters. That is, the moment the hydrogen ion enters the inside the cell, it will be sent out. The key cellular components like your DNA and ATP require neutrality, a neutral environment for replication, for functioning. So, for maintaining that in an acidic environment, for maintaining that neutrality, the hydrogen ion either should not enter or the moment it enters, it has to be sent out of the cell. So, that way the cell will keep on its membrane potential and survive. Okay. So, when most microorganisms grow in an acid media, their metabolic activity results in the medium or in the substrate becoming less acidic, whereas those which grow in the higher pH will have a lowering of the pH. So, the growth of a microorganism will alter the medium or the substrate's pH in such a way so as to support. The second aspect would be the effect of transport of nutrients. Normally, a bacterial cell has a negative charge, and because of this negative charge, non-ionized compounds can enter the cell, the ionized compounds cannot. Now, bring into the picture the pH. So, when the pH is either neutral or alkaline, organic acids will not enter the cell. Whereas when it is at acidic pH values, the organic acids become non-ionized and they will enter the charged cells. The ionic character of the side chain of the ionizable groups will be affected on either side of the neutrality. So, this in turn will result in increasing denaturation of the membrane and the transport enzymes. Okay. So, now what happens is that as the pH changes from neutrality, you are going to have more and more denaturation of the membrane proteins and the key transport enzymes. So, this in turn will disrupt how the transport of nutrients happens inside the cell. Then the third aspect here would be temperature. So, then the pH of the substrate becomes more acid when you increase the temperature. That is, the pH can change depending upon the temperature in which you are growing the food or you are processing the food. So, as temperature rises, you are going to become more acidity, it is going to become more acidic. Then concentration of the salt, that also will interfere with the pH. So, now what I have so adversely when you are talking about whether it is neutrality, whether it is alkalinity or whether it is acidity, this is going to affect the microbial membrane, the nutrient absorption and the key enzymes which are involved in the microbial metabolism. So, when any of these gets disrupted, the microbe will not be able to grow in the concerned food material. The next aspect is the moisture content. If we take the history of food preservation, drying or desiccation is the oldest method of preserving food. So, by drying or desiccation, we have removed the water content or the bound moisture which is present in the food. And without this moisture, the microorganisms will not be able to grow in the food, which means that microbes require water, a minimum amount of moisture content for their growth in a particular food. This water requirement of the microbe, we call it as the water activity, otherwise represented as AW, which is present in the environment. Now, how do we define water activity? This is defined as the ratio of water vapor pressure of the food substrate to the vapor pressure of pure water at the same temperature. So, at a constant temperature, the ratio of water vapor pressure of the food substrate to the vapor pressure of the pure water at this will define the water activity. So, we represented this as AW is equal to P by PO, where P is the vapor pressure of the solution and P0 is the vapor pressure of the solvent, which is usually water. And this is also related to relative humidity in the same way. Relative humidity will be 100 into the water activity. Now, pure water will have a water activity of 1. If it is alkaline with 22% sodium chloride solution, the AW would be 0.86. A saturated solution of sodium chloride has an AW of 0.75. So, when you consider pure water has 1, 22% solution is 0.86 and so saturated solution has 0.75. And in general, bacteria require higher water activity than fungi to grow. And among the bacteria, gram negative require a higher water activity than gram positives. So, by defining the moisture content or the AW value, we can restrict the growth of bacteria. You can control whether a gram negative bacterium is going to grow up or a gram positive organism is going to grow up by defining the or rechanging the AW of the particular food. So, remember we saw osmophilic, acidiuric, halophilic etc. in the case of organisms. The lowest reported AW value for bacteria is 0.75, which is for a halophilic, the salt loving. Then the dry or the xerophilic organisms, for example, the molds, and then you have the osmophilic organisms which can survive in high osmotic pressures, whereas the yeast will grow at AW values of 0.65 and 0.61 where the bacteria cannot grow. And at any temperature, when you lower the AW, the ability of the microorganisms to grow becomes reduced. So, by controlling the moisture content, you can inhibit bacterial growth, you can control the mold growth and modify the conditions in which the food is being preserved. And along with the AW, when you change the temperature, for example, the range of AW over which the growth occurs is great at optimum temperature of growth. So, you have a particular group of organism which grows at a specific temperature. you know the specific moisture content of the particular organism also. When you vary these two parameters together, you can control or restrict the microorganisms which grow at that particular moisture content and that particular optimum temperature. And also the type of nutrients which is present in the food. The presence of nutrients increases the range of AW, meaning if it is a nutrient rich food source, then the organisms tend to adapt in spite of the fact that the moisture content or the AW value would not be conducive. Because the nutrients are present in extra, the organism will tend to survive. And in general, the lowering of the AVO below the optimum is to increase the length of the lag phase and to decrease the growth rate in final population. So, when the lag phase is, the length or duration of the lag phase is increased, you are going to decrease the growth rate, size of the final population can be restricted, thereby reducing the overall microbiome load which is present in the particular food concerned. Now, the next aspect would be osmotic stress. So, to protect against the osmotic stress, microbes can accumulate what we call as solutes, compatible solutes. For example, the presence of potassium ions, glutamate, glutamine, proline, 7-aminobutyrate, alanine, glycine betaine, sucrose, trehalose, glucosylglycerol, etc. in the microbial metabolism can help the microbes to withstand osmotic stress. And two of these compounds, for example, glycine betaine, proline, etc. have also been proved to help the plants to survive osmotic stress. Now, in the case of gram negative organisms, they tend to accumulate proline. So, when you have an osmotic stress, gram negative organisms will accumulate more and more amount of protein by enhanced transport, meaning they increase the amount of transporters and they will start transporting more amount of proline into the cell metabolism so as to withstand the osmotic stress. In the case of halotolerant and zero tolerant fungi, they produce polyhydric alcohols like glycerol, erythritol, arabitol, etc. So, when these polyhydric alcohols concentration increase in the cellular cytoplasm, these organisms would be able to better withstand the osmotic stress and survive in the particular food concerned. The next factor which controls microbial growth is the oxidation reduction potential. Microorganisms display varying degrees of sensitivity to the oxidation reduction potential of the growth medium. This oxidation reduction potential is normally represented as O by R or the EH. Okay. Now, how do you define the oxidation reduction potential? It is generally the ease at which, how easily does the substrate lose or gain electron. Each organism has a potential to either accept or release electrons. This is what we call as the oxidation reduction potential, which is represented as O by R or the EH. So, when an element or a compound loses electron, the substrate gets oxidized. When the substrate gains electrons, it becomes reduced. Or in other words, a good reducing agent readily gives up electrons. Oxidizing agent will take up electron. So, when you have this electron transfer from one compound to other, whether it is a reducing agent or whether it is an oxidizing agent, you create a potential difference. This potential difference can be measured and expressed in millivolts. Now, if a substance is highly oxidized, then this electric potential would be positive. If a substance is more reduced, then it will have the more negative will be its electric potential. So, what is the electric potential of the particular food you are talking about? Is it positive? Is it negative? So, when this concentration of the oxidant and the reductant is equal, you create what you call as a zero electrical potential. And this electrical potential or the oxidation potential, which is expressed by EH OR, is different for different microorganisms and how they grow. For example, aerobic microorganisms require positive EH. For their growth, if the EH is negative, they will not grow. Anaerobes require negative value because they require a reduced environment. To summarize, in this particular lecture, we saw that three of the intrinsic factors which control microbial load, the pH, the moisture content and the osmotic stress. What are the different conditions in which the organisms can survive. In the next episode, we will talk about the rest of the intrinsic factors which control microbial growth in food. Thank you.